Okay, here we go, getting started on evolution of populations. So as we are talking about this, I want you to keep in the back of your mind what you saw in the butterfly lab that was about natural selection, and think about how it changed the population. Okay, all evolution means is we've got a change over time. Okay, so we're looking at a change over generations. You had 6 generations of butterflies, and every single one of you had a change in the frequency of certain colors in that population. Okay, so learning targets we're going to cover are 3 and 7. We're going to talk about genetic drift and gene flow, that's towards the end, and we're going to talk about mutations and evolution as well. Okay, so first of all, you need to understand that evolution does not occur on an individual basis. Okay, so you do not evolve genetically. Okay, populations are the smallest units that can evolve. So in the butterfly lab, your individual butterflies were not able to change their color so that they weren't getting eaten. But you can see this change in the population, that the butterflies that were able to blend in were able to reproduce more, and so you, by the end of the 6th generation, you had a lot more of 1 or 2, maybe even 3, depending on how your experiment turned out, of those colors. But you definitely had some colors that stood out and died off, or at least you should have. Alright, so natural selection acts on individuals. That's like the birds, or in the butterfly activity, it's like the birds that are eating the butterflies. Okay, but impact is shown when tracking the population. So you should see that. Think about the graph that you made. You can see the impact of natural selection. Natural selection acts on the individuals, but it affects the population over many generations. Okay, so population genetics, this is focuses on genetic change in a population, and it ties genetics together with Darwin. So it's really cool because Darwin came out with his book Origin of Species right before Mendel did, and it's cool to see how they work together. And it took a while for everything to kind of come together, but finally did it. It's pretty awesome. So if you take a look at these pictures of these finches down here, okay, these are all seed-eating finches that live in the Galapagos Islands. In 1977, this population of finches, they were devastated by a very long drought. Because of that long drought, they had a shortage of small, soft seeds. Okay, so birds with little beaks like this, during that drought, died of starvation because they couldn't break and crack into the harder seeds that were available. Okay, because when it's a long drought, a plant's not going to let go of, seeds if those seeds are just going to shrivel up and die. Whereas if their seeds are able to survive in a hard casing for until conditions improve, then the plant can still produce those. Okay, and so since those plants are still producing those harder seeds, these finches that have these bigger, deeper beaks, they're able to break and crack those casings on those seeds and eat the seed inside. So what happened is 1977, we have this long drought. These guys died. Since they died of starvation, not all of them, but a great majority of them, they died. And since they died, there weren't as many to reproduce. Think of your butterflies. You should have seen something similar like this in your lab. Okay, whereas these guys that had the bigger beaks, this one possibly, he's going to be kind of an in-betweener, but definitely these guys that have the bigger beaks, they're able to get some nutrition from these hard seeds, and since they're able to have those hard seeds, the next year what they found is that there were a lot more of these birds than there were of these birds, because these ones died of starvation. So you saw this in your butterfly example. Questions? Write them down so we can discuss them. Alright, gene pool. A gene pool is not a pool of denim genes. Okay, what it is it's all the genes that are shared by a breeding population. So if you take a look at my frogs over here, this is my frog population, and I have green frogs, blue frogs, and red frogs. So if I take a look at the genes that code for frog color, okay, you see I've got the green frogs, and I've got some blue frogs in here for the genes, and there's my red frog gene. Okay, all of those together, all the breeding individuals, that's my gene pool. Okay, then relative allele frequency, once again with the butterfly lab, you were able to, you actually found the frequency. That's what table 2 was. This is just the number of times that an allele occurs. It's expressed as a percentage, and its sum must add up to 1. That's going to be really important a little bit later. We'll come back to that, but you need to know that. Okay, then microevolution, what is it? Okay, microevolution is a change in relative frequency of alleles. So think of your butterflies. Some of you guys, I saw your greens were really successful, and so you had a change in the frequency of alleles from that 17% at the beginning to some of you guys were at 100%. Of the green butterflies at the end. Okay, that's a change in the frequency of alleles. It's microevolution. Okay, there's 2 causes: genetic drift, natural selection. So the lab we dealt with natural selection. Gene flow and mutation can also affect frequencies. Okay, so that mutations, there you go, that's kind of getting back to that learning target. How do mutations affect evolution? Okay, questions? Write them down. Gene flow, let's talk about that. This goes back to learning target number 3. Okay, it says recognize examples of gene drift and gene flow. So we're talking about gene flow right now. Okay, this occurs when fertile individuals, if an individual is fertile, it means that they can have offspring. Okay, so individuals that have offspring move into or out of a population. We have a flow of genetic information to wherever they go. So take a look at my population of birds here. Population A, it says selection pressure against recessive phenotype has created a homozygous population, big H, and I know that's cut off in the screen a little bit, that's why I'm writing it right there. Okay, so these birds, just like your butterflies, some colors stuck out more than the others. These birds have there's some kind of selection pressure that's killing the birds that have big H, little h, and little h, little h. Okay, so we have a higher frequency of this big H, big H. Okay, then take a look at population B. Same story, there's some kind of selection pressure that is giving us lots of birds with little h, little h. Okay, because it's killing off the big H, big H, and the big H, little h, whatever that selection pressure may be. Okay, and then by some occurrence, either the bird's able to fly around the mountain, gets caught in a storm and gets blown off track and finds its way on the other side, or whatever. This is just a very simple explanation, okay? Somehow one of these birds is able to leave population B and get over to population A. Okay, so this bird's genotype is going to be little h, right? Because that's what we're going to find in population B. Okay, so what happens when this little h mates with a big H, big H? Okay, set up your Punnett square. Please forgive my bad handwriting. It's difficult to write like this. Okay, big H, little h, big H, little h. Okay, so because of this gene flow, we go from having all big H, little h to now we're starting to get big H, little h in there again because we have this immigration from one population to the next. Okay, it reduces the differences between the populations. All right, here, sorry, you probably need that. I'd pause it because I'm going to move on. There's not much more to say. All right, questions? Write them down so that we can discuss them. All right, so sources of genetic variation. Mutations can occur during replication, those mistakes in replication that the DNA polymerase does not catch. They're rare. Remember, we talked about this. Okay, it's rare in our last unit. There's also gene shuffling, so this is going to be when genes kind of get all mixed up. And the source of gene shuffling is from meiosis. Remember, in meiosis you can have crossing over. That's where part of a chromosome breaks off and attaches to the other chromosome. They kind of just switch. They cross from one chromosome to the other. They switch spots. As well as law of independent assortment, all that fun stuff. That's all gene shuffling. Okay, this is the major source of variation. This is why it is so beneficial for organisms to reproduce sexually, because they go through meiosis and fertilization, which creates a lot of variation, which you could see if our population of butterflies was only one color, then and it was a color that stuck out, then that population is going to go extinct. Whereas since we had lots of different colors, that population still survived, but you saw how those frequencies of genes changed for those colors. Polygenic traits, these are controlled by many genes. In our notes we discussed skin color as an example. Okay, genetic drift. There's two things we're going to talk about with genetic drift. First one is Founder's effect. So chance occurrences lead to a shift in allele frequencies. Founder's effect, this is the founders or the original source of genes can have an unusual allele frequency, and then that's going to show up within the population because there's no new genes. Okay, so think of it as like founding fathers. If they came over, if they had some really unusual genes and the U.S. was completely isolated from the world, then we would pass all those unusual genes around the population. Okay, the frequencies are going to remain skewed if there's no gene flow, if there's no immigration. Okay, then there's also a bottleneck effect. This is when you have some environmental disaster that severely decimates the population. Okay, kills almost all, so it cuts off all but a few gene donors. So we go from having like, what are these? Little Pumbas. Okay, having lots and lots and lots of them with lots of different genotypes. Okay, and if we kill them off so we only have two left, those are the only two possible genotypes we're going to get. And what's the possible genotype of little b, and little b, little b? Well, it's all going to be little b, little b. So we've lost then that dominant allele from that example up there. Okay, the allele frequencies change dramatically. This is actually a huge problem that we see with cheetahs, and we can talk about that more in class. If you'd like to. Cheetahs were hunted very heavily, and now we've seen all of the variation from their population since they were hunted so heavily. They have very few gene donors left. Okay, so we run into lots of problems that way. Alright, so surviving population doesn't normally have the same genetic makeup as the original. Okay, we go from great diversity to small diversity. That's bottlenecking. Okay, selection on a single trait. Okay, there's selection for or against a single trait, and that's going to either increase or decrease the relative frequency. Just depends on the situation. So this here is an example of the peppered moth, and we can talk about it more in class because we're running out of time if you just remind me. But what happens is, just take a look. If you're a predator and you're in this kind of environment, which moth are you going to see first, the black one or the white one? Obviously the black one. So you're going to change, that black color is going to decrease in frequency because it stands out. Same kind of idea in this environment. The white one, though, stands out in that environment. Okay, so which will survive and pass its genes along, and I already talked about that. Okay, I think we will cover this on our next set of slides. Okay, so you're done. Please write down your questions and be prepared to discuss them as a class next time we meet.